In today's lecture, we will discuss about the structure of proteins. The sequence of a protein is determined by the DNA of the gene that encodes for the protein or that encodes a portion of the protein or multiple protein units. A change in the DNA sequence may lead to change in the amino acid sequence of the protein. Even if you change a single amino acid, that can change its overall structure and function. So to understand how a protein gets its final shape or conformation, we need to understand different levels of protein structure that includes primary structure, secondary structure, tertiary structure, and quaternary structure. So first of all, we will discuss about the primary structure. That primary structure is basically the simplest level of the protein structure, and that is simply the sequence of amino acids in a polypeptide chain. The hormone insulin has two polypeptide chains, A and B, and the sequence of A chain and the sequence of B chain can be considered as an example of primary structure. Secondary structure that refers to the local folded structure that forms within a polypeptide chain, that is because of the interactions between the atoms. And most common type of secondary structure are the alpha helix and beta pleated sheets. And both structures are held in shape by the hydrogen bonds, which forms within the carboxyl oxygen of the one amino acid and the hydrogen of the another amino acid. Tertiary structure, the overall three-dimensional structure of the polypeptide chain is called its tertiary structure. The tertiary structure is primarily due to the interactions between the R groups of the amino acid that make up the proteins. Important to tertiary structures are the hydrophobic interactions, in which amino acids with non-polar hydrophobic are grouped that clusters together on the inside of the protein, leaving the hydrophilic amino acids on the outside to interact with surrounding water molecules. Second important thing is the disulfide bonds, that is the covalent linkage between the sulfur-containing side chains of the cysteine, which are much stronger than other types of bonds. Last type of protein structure is the quaternary structure, that when multiple polypeptide bonds chains subunits come together, then the protein attains its quaternary structure. An example for quaternary structure is hemoglobin. The hemoglobin carries oxygen in the blood and is made up of 4 subunits, 2 each of the alpha and beta types. 2 chains are of alpha types and 2 chains are of beta types. This is the structure of hemoglobin that contains the quaternary structure. This is the overall level of the protein structure in single picture. Thank you.